Good evening, everybody. Today we will discuss CAFA competition going on Kaggle, and Sofya Trifonova will report us to the so-called HMMER. This is a tool in bioinformatics which might be useful for this competition to find the protein domains, and these domains might be used as a kind of features. So thank you very much, Sofya, that you agreed to make this talk, and please start. Oops.
Oh, thanks. Hello to everyone. Today we are discussing the program HMMER and possible application to CAFA5 competition. Let's firstly understand what are the domains in protein and why it is important for prediction of protein functions. The protein domains are the structural and functional units of proteins, and constituent domains primarily define protein functions. Also, domains could be described as folded and independent units in a protein. Anyway, these regions are highly conserved in a protein sequence, and you can see the illustration of domain organization on the right side of protein helicase DHR1, and you can see the protein sequence and the coordinates of each domain, and also you can see the positions of these domains in the structure of the protein. For example, domain reg A2, which is colored by dark green, is responsible for helicase activities in this protein. And there are many examples of such domain organizations. And this is just one example of it. Okay. And where can we searching for domains we're interested in? The main database for solving this problem is Interpro. You can find the complete documentation using this address. This picture from the documentation about Interpro, and you can see that there are many terms that you can find in this database. For example, families, repeats, active sites, and another. And also you can find here Go terms that used in CAFA5 competition. Okay. And how and where can we how we can find domains in proteins? There are many methods which can do this, but we are interested in the best one. And HMMER is HMMER belongs to one of the homology-based methods. There are some examples on. Oh my God. The internet, you know, not very good. Okay, you can find the examples of these methods on the left side of slide, and how it works. Domains are homologous segments in evolution, and also it can fold and work independently. So many methods work based on this information. The basic principle of homology-based methods is finding the homologous segments in different protein sequences through the alignment. But sometimes homologous template cannot be identified by simple alignment algorithms, and profiles can help in it because it can represent domain families rather than single protein sequences. And what is a profile? A profile describes the frequency of different amino acids at each position in a sequence that belongs to a given domain family. And what is HMMER and how it works? All information about this program you can find on this address. And HMMER is a popular package for sequence analysis written by Sean Eddy. It detects homology by comparing a profile HMM to either a single sequence or a database of sequences. And HMM is a hidden Markov model. Who doesn't know? Okay. A profile HMM is a rare variant of an HMM relating specifically to a biological sequence. Profile HMM turn a multiple sequence alignment, you can find it here, into a position-specific scoring system. It's like a position weight matrix. Profiles are statistical models that fix the evolutionary changes that have occurred in a set of related sequences. And the distinctive feature for it is using position-dependent gap penalties and substitution probabilities, which better reflect biological reality. And this profile consists of a linear set of match states labeled by M letters, and also there are also I and D states, I insertion and D deletion. And these states are connected by state transition probabilities, which also vary by position in the sequence alignment. And to sum up all this process, firstly, the program must obtain the multiple alignment. After that, the profile HMM is constructed, and this profile could be used to align this sequence and get a score. If we find the regions with a score that is higher than threshold, it means we find sequences with domains. And there are basic commands that could help you to construct profile and to find profile domains in protein sequences. All commands you can find on this address. So how we can, there are many details in the working of this program. I don't think that it's very important now. What is important, the application HMMER to CAFA5. Protein domains can be used as additions to embeddings. And also we try to understand the importance of these domains without generated embeddings, but it's still difficult to clearly understand how we can use it. And how I obtained the domains from train and test set of sequences, I used the command HMM search, and I used the profiles database PFAM-A. You can find it in the internet and download for your problems, for your tasks. And after that, I got the set of domains, but many of them belong to families. So I decided to filter them into only domains who found which is in PFAM database, and there are only 6,000 unique domains in both sets of sequences. And there are some problems we faced. Score is still low, and we can use some. We can use change some features that we can use for obtaining the higher score. For example, a number of domains in every protein, length of domains, or maybe filtering domains based on the occurrence and importance of functions. I think that is all I wanted to say, because this problem is not is open now. So that's all. You're welcome for your questions, for your suggestions.
Okay, Sofya, thank you very much. Nice presentation. So, colleagues, don't hesitate to ask the questions either in the chat or you can ask them by voice. So I see the first question in the chat. So Simon asks, how are the proteins distributed on domains? Are the group lengths similar? Similar, or are there some large clusters? Yeah, that's a nice question. So, Sofya, do I understand correctly? So now you have something like a matrix, which for each protein you have 0 and 1. Domain is found in this protein or not found.
Yes. Yeah.
Okay, and so can you comment on Simon's questions in the chat? So how some distribution, have you looked on something like a distribution?
I know that some proteins have many domains, some of them not many, and is the question about it. I don't know how many domains in each protein, but I know there are some different number of domains in each protein.
Yeah, what I meant was if there is some very general domains which maybe don't contain much information, or yeah, how I mean,
I understand.
Yeah,
it's very good idea to search for importance of each domain, but I didn't. I don't know yet this, but I plan to do it. Last weekend. Yeah, it is right. And we should to understand which we can use for obtaining the higher score.
There is another question, Sonia. So first, thank you for the presentation. Do you think it would be worth looking into linker regions as well as domain regions? What do you think? So the regions of the proteins between the domains?
I think it's not very. I think these regions are not very important, because domain regions are responsible for functions, and we are interested in functions. We want to predict functions in the in proteins. So linker is not very it's not very important.
Okay, Thank you. Colleagues, any other questions, suggestions, comments, whatever? Eden, maybe you can comment on this.
Yeah. So a linker, so there's a little bit of confusion here between, and a lot of people do that, I'm not blaming anybody, between a domain and a conserved sequence and so on and so forth. So a domain, we might say that a domain is probably a large region of a protein, and structural biologists tend to say that domain is an autonomously folding unit, meaning something that if you disconnect from the rest of the protein will still fold more or less into the same shape, which, you know, may or may not be a good definition. But and then you have the smaller areas that are like, you know, conserved sequence areas and conserved sequence region and so on, that are so a domain might be, you know, 100 or 150 amino acids. A conserved region can be, you know, 10, but still has kind of a very important structural feature. Now, linker regions between domains, between two, you know, these are my thumbs, that's a linker region. The domain would go like this, is actually important functionally because, hey, my protein goes like this, and this might be important for me to understand the function, right? The other thing is that disordered regions in the protein, which linkers usually are, can be important because they might actually bind another protein. And knowing that they bind another protein, that may be a specific way, is also, can also be a little bit of important information. So it's a question of where you are looking, right? More than that. Now, if we want to concentrate only on domains or only on, you know, say, conserved regions, then like I said before, you know, some of them contain important information. So, for example, if I find a DNA binding region, oh, that's important. I know this protein binds DNA, and that narrows down its functionality quite a bit for me, right? Or it may just have something really incredibly general, like a transmembrane domain. Okay, I know it's in the membrane. That's actually embedded in a membrane. It's actually pretty important too, but doesn't tell me as much, right? So these things are I guess I'd like to say everything's important, but of course we need to know where to focus.
Thank you very much. Any other questions, suggestions, comments, whatever? Sofya, can you go to the previous slide? So where you used this HMMER search. So for this, so this domain. So have you taken these domains from some database? Yes, this is correct.
This is correct, yeah.
But the database, I think it's, as far as I understand, it contains more, something like, I don't know, 20,000 domains, yes? But you have only 6,000 here, so that means not all the domains have been found here, at least with, is it correct? Do I understand correctly? Or all the domains from this database have been found?
This database is consisted from profiles that describes domains, yeah. But you know, I think you're right, because there are more than 6,000 domains, but I used the train set, and this train set is doesn't include the all proteins, so it's logical that there are not all domains are found, just only 6,000. And it's okay, it's normal.
Okay, and the technical question. So, how long time the execution of this have taken? So,
not very. It was not very long process. How long this program
yeah.
worked,
So we need
was working.
Yeah, so we need more than 200,000 proteins. We should align to these domains.
I think about about 20 minutes, maybe 15 minutes.
20 minutes, okay. And so you made it on your own computer or on Kaggle? It's also possible to.
I tried to do this in the Kaggle, but there's many. I couldn't do this in the Kaggle, so I just I did it on my computer.
Yeah, sometimes it's difficult to install some packages on Kaggle. Yeah, it's a pity. Okay, thank you. Colleagues, any other questions, suggestions, comments, whatever?
I can clarify something maybe about the difference between domains, families, and so on. It's actually on
Yeah, thank you.
the, if I can share my screen. Oh, just a minute. Somebody has a question.
There is a question, just a moment. So Simon asks, maybe one could try to train very simple model for few epochs using one of the standard embeddings and see how well it does in predicting domains. This way we can see how much of the domain information is captured by the large language models. Yeah, that's a nice idea. Well, in Simon, in the announcement of this talk, I put a link to the Kaggle dataset, which was from the Google AI, and they something like so. As far as I understand, the task was something like that.
Okay. Sorry, I didn't see. Yeah, I mean, I was thinking. Okay, ESM3 is an LMM that does. Yeah, I mean, I was just thinking that maybe one could also try to, you know, fine-tune a model on this task and then remove the head and then go on with the protein function prediction. So maybe there's also a way to go, but
very interesting, yeah, idea. Yeah. There is another question on comments. So thank you very much, Sofia. I have a question. It's not about CAFA5, but about application of the HMMER in taxonomic classification of metagenomic data. Could you tell, please, what database is better to use for it to detect remote homologs? Sofia, can you comment on that question? It's in the chat.
I can see this. Okay, but I think that Interpro could convene for this because there are many things in this database. If I don't mistaken.
Eden, maybe you can comment. So I see you write that HMMER is not good for that.
Sorry, I'm muted. I believe that, well, for one thing, HMMER is for, HMMs are good only for long sequences, okay? Now, metagenomic sequences are very short reads, and they're DNA, and HMMER is mostly a protein. I mean, HMMER can be used for DNA, but not really. Or rather, it's not the intention. So short sequences and DNA are not like long sequences and proteins. So HMMER is not. There's a whole different kettle of set of tools to taxonomically assign metagenomic data. Kraken, Kaiju, for some reason, they're all named after monsters. But these are the kind of thing. I don't think that's really on. Yes, you can assemble contigs. That's a problem because you might get chimeras, meaning you're assembling contigs from different organisms because it's metagenomic data, it might come from different. So that's one problem. Extract an ORF. Yes, you can do that. There's a whole bunch of stuff there, but really, I think this conversation is a little bit off topic for CAFA5 maybe. I mean, it's an interesting conversation. I'm not trying to be dismissive of the ideas, but it's a little bit off topic, and assembly of metagenomic data is a whole different kettle of fish because, like I said, you might get chimeras, you might get, in many cases, you get contigs from different organisms together. So there's a whole question of binning the contigs without assembling them. Kraken and Kaiju are the ones you want to look at for that. Not Kraken, sorry.
Okay, thank you very much. So Okay. Either you want to make some comments on the domains?
Yeah, let me just try. I lost my screen again as usual. Can I share screen?
Yes. So, Sofia, can you please stop sharing and then?
Oh, okay.
All right. Disable annotation for others, right? Here we go. So this is, so PFAM got incorporated into Interpro. Just saying, so now this is it. And you can see that here we have, so it's funny because you actually have a mapping from PFAM to Interpro, from PFAM domains to Interpro domains and so forth. Just a minute. And for some reason, I got the browse by entry of Interpro here. It's getting very confusing now. But the general idea, okay, let me go to another screen here that I prepared. So here I tried to search Interpro by a PFAM domain, an Interpro domain, and I added another domain. So I added two domains here. One is zinc finger and one is bzip. And I found this protein that has these two short, I guess, domains here. One and it's really beginning to be a little bit all the same. They're kind of all mashed up. Oh, PFAM. All right, here it is. Now it's in PFAM. Okay. So this is an zinc finger-like. And when I go to the PFAM family, as it were, it describes what type it is and what it does. And zinc finger in this case is a DNA binding. So that's kind of, again, one of those important things. I want to kind of make sure people understand also the idea of the hidden Markov model. So if we take a multiple sequence alignment, we can just build kind of a regex-like thing from it. If you can see this in multiple sequence alignment, glycine is all the way down, histidine is all the way down, glutamate is all the way down. Here we have a little bit of a change. So this is marked as X twice. Then we have a conserved G here again. We have a mostly conserved G here again. But it's kind of hard to capture the fact that here we have a conserved G 100% and here we have conserved G 60%. So a matrix that gives the count of the amino acid versus the length of the alignment is also a good thing. And the hidden Markov model is probably the best. Where's the hidden Markov model? And the hidden Markov model is probably the best way of doing it because it doesn't just give a matrix, it gives a look back. It gives a probability of what goes on. Given that the previous three were this, and this, what is the probability of this being? So a matrix is kind of a position-specific score matrix, kind of a zero look-back Markov model. So those are the differences. And yes, HMMER and HMM scan, which PFAM uses, are probably the most sensitive way to look for similar sequences based on homology, based on similarity, right? The similarity means that they are similar and it's very sensitive. But once we discovered embeddings, this whole similarity entails functionality. I wouldn't say goes out the window, it still exists, but we can find a lot more similarities without any apparent, we can find more functional similarities without apparent sequence similarities because the embeddings capture them, right? So it's like in natural language, when we find, the words Paris and Rome are not similar to each other in any way, but they both convey the functionality of being capitals of their respective countries. So the same way with embeddings, when you have two embeddings that are functionally similar, if you train your model, you can find, yes, they are functionally similar, even though there's no sequence similarity, simply because the similarity is somewhere else, usually in the biological process rather than molecular function. Molecular function homology still is a good capture, but biological process, that's where the embeddings really gave us a huge kick upwards in terms of predictive ability. So I'm not dismissing. I mean, of course, I've been doing HMM and sequence similarity most of my life. Embeddings only came to be, 4 years ago, useful in this field. But I just like to put these things kind of a little bit in maybe historical context, and of course, in Markov models and similarity based is still incredibly useful.
Thank you very much, Ida, for your comments and explanations. Colleagues, so any other questions, suggestions, comments, whatever? Maybe to Ida or to Sofya. So don't hesitate to ask questions or comments. Okay, may I ask a question? So but in general, what do you think about this pipeline that we take these proteins and make this alignment to the FAM database? It's a kind of standard pipeline, or it's kind of not the best way to do it?
Standard may be a good way of doing things sometimes. They don't. The two are not necessarily mutually exclusive. I must say that when she was in the business bit, I think I missed a couple of slides because somebody knocked at my door and I had to get up. That's when I put myself on out of the camera. So I may have missed a couple of things here. No, I think it's good. I'm just wondering, I'm a little bit rusty on my PFAM thing, especially since they moved to Interpro. I think that PFAM uses a different HMM aligner called HMM scan. I'm not sure. And maybe that they use HMMER. I see that Interpro also incorporated HMMER. Interpro incorporates everything, apparently. So yeah, HMMER. I mean, in HMMER, I think you have two stages, right? And again, I missed your pipeline. One, when you compile your multiple sequence alignment into a hidden Markov model profile. Is that correct, Sofya? And then the second one in which you search.
Sofya, can you comment?
I'm sure it is correct. I used the command HMM search. This command works like the HMM scan, and this is faster than HMM scan. And I used the database with profile, and I don't understand the question.
No, maybe I was getting a little bit too technical. I apologize. Mike, I guess my question is, when you took the sequences, they were already against what you're scanning, your sequence database. Did you construct it yourself from scratch from sequences, or did you download like an already a set of pre-constructed HMMs? PFAM-A, for example, is already pre-built HMMs.
I used just downloaded database and the set of sequences of train and test sequences. I didn't do anything with these sequences because I didn't need it. I didn't construct any alignments. Just used the set of sequences and the database with these profiles. Yes.
Mm-hmm.
And it works properly, so everything is all right.
Yeah. Okay. No. Great. I was just wondering. No, I don't have any.
Okay.
I mean, HMMs have been around for a while now. Some of the more successful methods in CAFA have been using, variations on HMMs, including using HMMER, I believe. And like I said, this is mostly good for jacking up your score in the molecular function ontology. Biological process homology based is not as great as detecting. The reason being that if you have several proteins in the same pathway, they usually are not homologous. So you can find the specific enzyme that does something there, but you may miss out on another protein that is totally not homologous in the same process that does something. So that's where actually language models can come in to be somewhat more useful. And genomic context models. So these are models, maybe something we'll talk about next time. Models in which you look at genes along the chromosome. Say we have a conserved region in one organism, ABC, and in a second organism also genes ABC, always together, ABC. Why are they always together? They're probably working together, right? So, and suddenly you find an ABD, but no C in the genome. That means that D replaced C as what is known as a non-homologous replacement. So these kind of things have proven to be more useful in upping the score for biological process. So homology can only go so far in certain aspects, but it's still very powerful. So I applaud this actually.
Okay, thank you very much. And another question maybe either to you. So if kind of with this protein domains approach, maybe we can get something like more interpretable models. For example, if we can connect that if there is this domain, then probably this protein is related to these gene ontology functions. Would it be interesting, or it's a well-known kind of, what's your comments on this?
So the way PFAM is currently set up is that, yes, that's exactly what they're doing. They're mapping domains to gene ontology functions, right? That's the idea. They're mapping domains rather than mapping proteins. So if you look at, and that's probably important to say, so thanks for bringing that up. So if you go to, say, the GOA database, the gene ontology annotation database, and you look at, say, the entries of proteins there, you have several entries per protein, several functions for the protein, right? Like, because it may have several functions, even in the same ontology. And those functions are carried out, say, by different domains. Now, the GOA file will not tell you that. The gene ontology annotation file will not tell you that the mapping of the domains to functions. However, if you go to PFAM or HMMER, well, HMMER is the search engine, PFAM is the database. If you go to PFAM, that will tell you that. And PFAM domains are associated with gene ontology annotation. So it's a finer mapping, to the specific positions in the protein. That now CAFA doesn't capture that. Gene ontology annotation. We talked a lot about how whether and if CAFA should capture it, and we did play around a little bit with the idea of doing domain-based annotation rather than protein-based annotation. But it was hard to it's hard to run an assessment on these things, I believe. Frankly, I don't remember why we dropped the idea. Maybe we should revisit it.
Okay. Okay. Thank you. Colleagues, any other questions, comments, suggestions?
Sorry, can maybe someone show how to find PFM and function? How to look this PFM, do this function? Where on web?
Luda, you mean that this database farm where exactly it is stored the correspondence? Or?
Yes.
Yeah, I can show that. I think that it's in the PFAM. I think if you download PFAM, where can I share my screen here? So go to the download page, PFAM tab. you have the models here. And I believe those are mapped to Go. If you download this, the PFAM-A models. And if not, the gene ontology database, Go itself will have, so from the other side, Go to Go, pardon the pun, and it'll have a mapping to PFAM. Somewhere there is a mapping, or you can just Google it maybe. But there is definitely a mapping of PFAM to Go mapping. Yeah, there is definitely somewhere. Oh yeah, haha, found it. There's something called PFAM to Go. Here it is.
It's simple. Okay.
Yeah, but it may not. You have to look. It may not be very reliable. This is from. Oh, this is actually pretty new. And this is from gene ontology, so it's probably pretty. So here you have the PFAM domain, and here you have the Go.
Wow.
Okay.
Yeah.
All right, you have a whole text file here.
Is it means that this 600 domain we can directly map to function?
Not all of them. Well, I have a lot more. I mean, these are PFAM. These are PFAM entries in the database. Not all PFAM entries are PFAM domains. Some of them are families, meaning whole, whole proteins, not just domains. Some of them are smaller. They're like sequences, small sequences. Okay, so these are PFAM entries. Not all PFAM entries are domains. So you'll need to filter out those PFAM entries that are domains, and you can do that from information from the PFAM database. The other thing is not all domains, their functions are known. There are domains called DUFs, DUFs, domains of unknown function. There's actually a big project now mapping the DUFs of the human genome that is going on in, forgot somewhere. So that is something that you can look into. So not all PFAM entries are domains, and not all domains are associated with functions.
But if this is linked, how often it's true?
How often? What is true?
It's true. It's true information if I see domain and function.
Oh, so how trustworthy? How much can you trust the information? Good question. There's a paper here. I guess we have to read it. On how they did that exactly, because we talked a lot. I think the last meeting, there was a lot of talk about how trustworthy the information is in terms of evidence codes, right? And the evidence code, they're experimental and non-experimental, and what is trustworthy and what isn't. And there's a whole story about that. Frankly, I don't know. I think you'll have to dig a little bit deeper and see how exactly they did this mapping, right? How was this mapping done, right? So, sorry.
Maybe you know why only so little protein in Swiss Pro? How protein becomes Swiss Pro from?
Why there are so few proteins in Swiss Prot as opposed to proteins in Tremble in general? Yeah, okay. That's because we don't have enough biocurators. So everything in Swiss Prot is reviewed. Okay, some human had an eye on it. Maybe not for very long, but they had an eye on it, and that is what makes Swiss Prot what it is. That's the criteria of inserting into Swiss Prot. Now, to get into Tremble, you don't need to do much. You just need to have a genomic sequences that probably translates into a protein. And once it's there, it goes into Tremble. It may not even be a real protein. And we've seen actually, I think recently, an example here in CAFA that one protein got removed from the database after we compiled the training superset, right? Because it was considered not to be a correct protein. It's considered not to, or it was a duplicate or something. So, so Tremble is an unreviewed set, and it's just, I mean, it's important, but it's not being reviewed by people.
May I ask you another question? So, approximately, how many proteins they go for example, in 1 year from Tremble to Swiss Prot? What's the capacity of this curation? Approximately, just.
I knew the answer once. I think we looked at it in one of our papers, but I don't remember. A few thousand, I believe. Let's see. Yeah, a few thousand. And it kind of depends also on an effort that's going on, because it's not like there's this group of people that are sitting in one room and doing it, right? It's a bunch of labs all around the world that are annotating. So let's say suddenly, I don't know, some Drosophila group got a lot of money for biocuration or for running experiments or for both on a fly, and they gather a lot of data there. So suddenly there's a whole influx of fly genes coming in. And one of the CAFAs, I think we had 400 Arabidopsis gene drop in one day. You know, so it kind of goes by leaps and bounds, by interests, by there was actually a really cool paper that somebody wrote a few years ago about how to predict what biocurators will do next, and therefore you can win CAFA that way. They didn't do very well in CAFA, but they did pretty well, but they weren't too shabby. So that was another strategy.
What
Okay.
is biocurator?
A biocurator is a person who basically provides the functional information for a protein. So let's say you have a paper on a certain protein, the biocurator reads it, interprets the information in terms of the ontologies that are there. So assigns ontologies to the protein based on the paper and does that. So that's where you see, say, the experimental evidence that is coming in. Somebody read a paper and put the data there, and also gave the PMID of the paper so you can read it and see what they did. So these are persons that functionally annotate the proteins. Those are biocurators.
It's a more function, it's a new function when people review annotation, or it's often drop some function. What's change when people?
Well, it's usually new functions. You're right that sometimes there are corrections, but not as many. It's not necessarily new functions. You might have proteins that are, you know, it might be that somebody came across a paper from 2003 about a protein and said, oh, wow, this is a new thing. We haven't, I mean, it's new to us now. We haven't annotated that protein as such. Let's say you have a protein, and sometimes it may not even matter that much. Sometimes it does. So let's say you have a protein that has 99% identity to another protein, A and B. A is someone, no literature on it. B has very strong literature on it. They're 99% identical. You can pretty much say that they do the same function regardless. This is pretty trustworthy. However, suddenly for paper A, somebody comes across for protein A, somebody comes across a paper from 20 years ago saying, oh, it also does this function. Nobody knew that. Or people actually knew that, but it was never captured by the database. There's a question also of knowing and the question of being captured by the database. And the lag here is always a problem also.
You know, and what is the kind of, how big is this lag? What's your kind of feelings?
It depends. It's usually a couple of years, but it may be, I mean, we've seen 10 and 15 years sometimes, but usually not in critical issues. Sometimes the lag, sometimes there is no, sometimes the lag is like a couple of months, especially in hot research topics. Sometimes there's never catching up, especially in proteins that are, you know, are not deemed in to be such interesting for research, or that have this 99% identity to a protein that is known. So why bother? Things like that.
And another question about this 99, as you say, or kind of 90 or some. When I looked in the CAFA data, current CAFA data, I see this kind of problem like that. I see some proteins which are very similar, but their functions are quite annotated in the current CAFA, quite different. And I feel this is a kind of maybe one of the main problems for the machine learning.
Talk to my, I have a student here. Oh, Henry is here. I have a student, Parnal. She's actually researches this very thing. And that has to do, we think, sometimes with the fact that in different organisms, there are different interests and different abilities to research function. So let's say we actually discovered that there's quite a bit of difference in between mice and human, for example, in the annotations for proteins that are orthologs or proteins that are homologs. And it's not because the functions are different, it's because what we looked for in mice and what we looked for in human is different in a way. And that doesn't mean that they don't do the same thing in human. There are experiments we can do in mice that we can't do in human, right? That's the whole idea of a model organism. So we have these assays that we do in mice, and we find dysfunction in mice, and then we say, okay, well, mice are evolutionary close, so to speak, to human. Therefore, we transfer that function to human. That's a whole philosophy of model organisms. But we don't see that annotation in human because it was never annotated as such in human because you couldn't run that experiment on humans, right? You ran it on mice. So that is one source of such differences.
And the kind of what to do with that, because it creates a problems for the machine learning, because you have similar proteins, but the labeling is different, and machine learning gets crazy with this.
So there are several ways. One is set yourself a standard. You can, okay, for one thing, yeah, this is a hard problem, but notice that the protein function prediction problem is easy, right?
Yeah.
A lot of things about it are really difficult.
But may I ask the following? So
yeah, add organism information. That's important. That's almost as Andre just told me directly. Add organism information. That is added. That's correct. That'll help us look at organism-based bias, which is something we are looking at. Another thing is saying, okay, if we trust this 99% identity towards function, we just percolate or propagate, not propagate, propagate is up to 3, so I won't use that term. We just transfer the function from mouse to human based on this trust. And that is the whole philosophy of something called the paint initiative in Go. I'll put that here. That to do these things, propagating across the evolutionary tree. And I'll just put that here. And you can look into paint. Paint is the idea to propagate information across the evolution tree based on a certain standard of trust that is based on sequence similarity and closeness on the evolutionary tree. So if you trust mice to be mostly human in that respect, which you may or may not agree with, then you can do the paint propagation.
Okay. Thank you very much. And another question is, so what will be on the kind of private data set, so the final kind of evaluation? Because so is it correct that, not all the functions will be experimentally tested for these proteins. So maybe our kind of predictions will predict something which is correct, but there will be no experimental check for this. And so the evaluation will kind of say, give us a penalty for that.
Yeah, so you're worried about what we call the open world problems,
Yeah.
or the missing information problem, meaning that you may predict a true positive, which is in real life a true positive, but we will see it as false positive because that has not been annotated yet.
Yeah.
So 2 answers, right? The first one is that, yes, it's a problem and you may be penalized for that, quote unquote, of course. We do, for standard CAFA competitors, not the Kaggle prize money one-shot thing, we actually redo CAFA every once in a while on new sets and find out. The other thing is that it doesn't affect the score that much, surprisingly. And we actually wrote a paper about that. I can put that in. It doesn't affect the score as much for reasons I won't get into. So don't be afraid to make aggressive predictions. It's better than to overpredict than to underpredict in the way we're scoring things.
Okay, thank you. And so just to understand this experimental evaluation, so if we have a protein, yes, and you make some experiments, so you check experimentally some properties, but we have something like 40,000 gene ontology terms, so it's impossible to check them in kind of one experiment. So what is kind of your estimation or your feeling? What is the percent of the correct these annotations will be experimentally set it as kind of true positive?
So
And what will be a kind of open world problem?
So in previous CAFAs, so are you asking me how many experimental annotations do we expect to get over the waiting period, like between August and December? How much additional information we expect to get? Is that what you're asking? Or no?
I'm more about this open world problem. So you will have some protein for the for the final evaluation. So you'll make some experiments which will check some gene ontology terms. But due to these open world problems, there can be something which you will not check experimentally. So what is your feeling? This will not be much. This will not much affect this evaluation.
It won't affect much, no. And just a minute, I put the wrong paper up there. You can read that. It's an interesting paper on one of our scoring.
Okay.
But actually, this paper, open world problem. So there was a paper written exactly about this problem by Jesse Gillis. We cite him here, and this is actually our kind of a reply paper. What is exactly your question, Alex? What is the effect of that? How about the effect? And we show here that there is some effect, but it's not very large, and you can read, and it kind of depends also on the metric you use for assessment, really. And you can read it in this paper.
Okay, thank you very much. Yeah,
Sure.
I'll read it. Thank you.
It's the impact of incomplete knowledge and the evaluation of protein function prediction, structured output learning perspective. So, yeah.
Excuse me, I have one further question, if I understand correctly. So we can, for example, compute sequence similarity pairwise with the diamonds. And then what we could do is to set us a threshold of 99% and say, okay, if we have 99%, we take all the Go annotations and spread them over this cluster. I mean, how can we set this threshold, or is this software what you pointed to doing this, or in a more?
Yeah, so it's more like a methodology than a software. And I'll put the exact answer to your question here right now. I just put it in the chat. So this is making an inference, transferring annotation. When can you transfer annotation, when not? It doesn't have to do necessarily just with similarity, although 99% is probably pretty safe. It has to do also with evolutionary relationships. So it's not fully captured by percent identity. It's something you want to actually look at a tree for that. So I suggest you read this Wikipedia entry on Paint, on the phylogenetic annotation and inference tool. This will give you an idea of transferability of annotations.
Thank you.
Sorry, can you look if it's a big function annotation, which one is more interesting now? Which one is grow a lot now? Which is maybe some old function and some is interesting now?
Yeah, good question. So there's fashion in science, right? Like in any other human endeavor. So for some reason, viral proteins have become very popular recently. I'm not sure what happened in the world that triggered that, but that has become incredibly popular. Specifically, something called the spike protein. I don't know if you've heard about it. So that has become popular. And again, it's kind of driven by health and need and interest. Cancer is always interesting. It's considered to be a disease that strongly affects millions of people worldwide, and is especially impactful in developed countries. And those are the countries that have the money to do that research, right? let's say that, for example, malaria was less interesting, even though it has a huge impact, but it has a huge impact in, those countries that can't afford that much of research. It doesn't mean that there isn't a lot of research going to malaria. It just means that there's pound for pound in terms of impact, there's less that than in cancer. There's a lot of research going on too about how research funding is allocated across different endeavors based on human interest and so on. One interesting initiative that was done by the NIH about almost 10 years ago was trying to find proteins that are associated with highly researched proteins. So you have proteins that are highly researched, and for some reason, say they call the druggable genome, those that are associated with internalizing drugs into cells. And those are really heavily researched and so on. And then they said, okay, well, these proteins are also, looking at, protein binding interactions and stuff like that we see all these time in the database in CAFA. Maybe there are proteins that interact with them that we haven't looked at yet, and those would be interesting to research. And there was a whole idea of elevating the interacting proteins that are under research to being researched more. And that's another strategy that's going on. So I know I didn't answer a question about what is being interesting and what is not. I'm just saying how these kind of research dynamic work. Or one thing that we found is that, for example, sometimes it's driven by technology. So there was this technology event 15 years ago, maybe, developed to look into embryonic development in worm, in C. elegans. It's a type of a worm. And suddenly everybody was using it. A lot of papers came out. And if you looked at C. elegans, you could think that half of the protein C. elegans, all they do is involved in embryonic development. No, they're not. It's just where people were looking at, right? So they were looking under the street light. So the secret being a technology to give you something.
Yeah, very interesting. Thank you very much. Colleagues, any other questions, comments, suggestions?
Sorry, if we look at one protein, we can find all his function, or only that function that we knew we look for. We find only that function when that we predict we can find and we look for them, or we find all function of protein.
I'm not sure I understand the question.
So what is the role of the prediction? So can you, if prediction
not
doesn't
prediction,
tell you this
what role of intuition? Do you need intuition for look for some property in protein, or you take protein and you can find all its property?
To make an experiment, can you make an experiment to check all these 40,000 of the gene ontology terms in theory?
Yes.
Theory.
Yes. Or you look for one term.
Or it's
usually, yeah. So usually you run an experiment. Yeah, usually an experiment checks for a binary prediction, right? And the binary is a function or a not function. So it takes tests for one functional. Some experiments, some assays can test for more. So for example, it kind of depends also your definition of function again. So for example, there's this, you can run experiments for protein binding and say have this assay of a bunch of test tubes, say a cluster of 100 test tubes altogether. Each one has a slightly different protein in it, and you take your own protein and chuck it into those test tubes. I'm really murdering the science here, but the general idea is that if it binds, there's some reaction that goes on the test tube that changes color or whatever, and then you see, okay, this protein binds A, B, C, and D. So now you have 4 different functions: binding A, binding B, binding C, binding D. Or you have one function. This is a protein binding. Okay, so that's one question. Or another experiment is you want to test whether this protein, so you know it's an enzyme and that it catalyzes a certain, I don't know, breaks up a certain type of sugar, but you want to see whether it breaks up other types of sugars as well that are chemically close to this sugar. So you take all the different isomers of a certain sugar, you put them in the different test tubes, and then you put that enzyme in and see whether it catalyzes those sugars or not. So it's this sugarase enzyme. And now you kind of refine the spectrum of what you're looking for in terms of the types of sugar this sugarase interacts with. And this is not, and this is a little bit, this is where the whole ontology things gets messy, because it might do so, but with different efficiency. So if it breaks up a sugar really, really slow and inefficient, and only like 20% of the sugar is being broken up, and does it really do the job well enough to warrant a gene ontology annotation? And gene ontology doesn't capture that inefficiency in functionality, right? So how good you are at what you do, gene ontology doesn't capture that. And that might actually be important, right? You, as a biochemist, I might want to know whether this is an efficient enzyme or inefficient enzyme. I may want it to be efficient or not.
This efficiency, it writes somewhere that this is do this function good and this is a little.
Yeah, I mean, you can annotate that. I mean, there's a whole field of biochemistry and enzymology that talks about, you know, quantitative enzymology and how well things break up and so on. It's kind of first year biochemistry.
And
I have
to
leave now. I'm sorry, I'm late for my group meeting, so I apologize. I have to go.
I think, yeah, thank you very much, Ida, and thank you very much.
Okay.
So I think
I'll
maybe
leave.
for
Thank
today.
you. Great questions. I'll try to make it. There's another meeting Monday, right?
Yeah, so we plan on the Monday evening, yes, another meeting. I'll make an announcement later.
I'll try to
And
make it.
if someone wants to make a talk or suggest some reports, please, you are very welcome.
Okay, talk to you later. Thank you, everybody.
Thank you very much. Thank you very much, colleagues. So, Sofia, thank you very much again for the presentation. So I think we can stop here and maybe continue discussions on Monday. And so thank you very much, all the participants, for your participation. The video recording will be soon on YouTube, maybe tomorrow. And so goodbye, everybody. Thank you very much again.